Okay, so like I just said, saltatory conduction is when action potentials are jumping between these adjacent nodes of ROM-VA. Now, the question obviously follows, why in the world would you want to do that? And the reason that this is a good thing for neurons to do is that it increases the conduction velocity. So if you want to move a signal very quickly down your axon, if you're having this saltatory conduction, you can move this signal down your axon at a much quicker rate. Well, why is that? And the reason this is that ions crossing the membrane, so when ions come into the cell, this is relatively slow, and that's because there's a limited number of channels in the cell membrane. They're not infinite. And so this is going to be relatively slow compared to ions diffusing down their electrical gradient inside the cell. This is quite a bit faster because they don't have to find an ion channel to make its way through. It can just diffuse through the cytoplasm. So this is a much quicker action than what it is when they're just coming in. So let's kind of take a look at an example of this. What we have here is a cell. This blue stuff up here is a myelination, or a strip of myelin right here. The red kind of half circle here, or this red half tube, this is an axon. And just for simplicity's sake, instead of showing all the ions and all of the channels, I'm just going to show pluses and minuses here, and showing pluses just wandering into the cell. You know that the pluses, these are going to be sodium ions moving into the neuron. So let's go ahead and take a look at this. So what we have here is one node here, and then what we have over here is the second node. And let's go ahead and take a look at what happens here over this. Well, these sodium ions start moving in, and they move in relatively slow, but then because when they move in, there's going to be this area where it's essentially electrically neutral or close to neutral. They're going to diffuse very quickly to the nearest area where it's not neutral, where it's negative, because that's down their electrochemical gradient. That's going to depolarize that area of the membrane and cause more positive or more sodium channels to open up and come on in. But when this nearest negative charge is really close, they don't move very far. And so we actually have this signal moving down the membrane at a pretty close or a pretty slow rate. But watch what happens when it gets to the end. This last bit of charges is going to jump very rapidly to the next node and start to depolarize that. And so if you have more of this myelination, more of these nodes and these areas in between they're myelinated, we can have this saltatory conduction where these depolarizations are going to move very quickly down the myelinated area and start depolarizing the next node. And this is going to increase the conduction velocity sometimes up to 50 times. So myelinated neuron axons are conducting at velocities about 50 times greater than their unmyelinated equivalents. And by equivalents, I mean basically the same size. So the functions of myelin and saltatory conduction are a few different things. First off, this is preventing leaking of charge out of the cell. And if you remember back to when we were talking about graded potentials and passive conduction, well, if you can increase the resistance of the membrane and prevent these charges from leaking out of the cell, this is going to increase the possible distance that you can move this passive conduction. So this passive conduction is really fast. So we want to use it as much as we possibly can. We want as long distances as we can get out of this passive conduction. And what that means is if we myelinate or insulate these axons, that we can get kind of the most bang for our buck out of this passive conduction. We can make it go as far as we possibly can. Also, this prevents a potential from forming between these nodes. So this is also another way of separating charges so we don't get this potential being set up along this membrane, which would blunt the ability of these ions to move down their electrochemical gradient. Another really big thing, especially with animals that have lots of nervous tissue, like humans, is that this is going to conserve a lot of energy. And the reason for that is that you only have to run your sodium, potassium, ATPases now at these nodes of ROM-VA. You don't have to run them the entire way along your axon to keep all these sodium-potassium ATPases working the entire time. So what we're actually getting is we're getting higher conduction velocity out of less energy usage. So that's a really big plus. However, that's not to say there is no disadvantages to myelin. Now, it actually turns out that myelin is really expensive to make. And expensive not in terms of energy necessarily, but in terms of materials. Myelin requires a lot of lipids. As you remember, we're taking a cell membrane and wrapping it many, many times, sometimes up to 200 times around this axon. And this is going to require not just lipids, but one very expensive lipid, which is cholesterol. And there's a lot of animals out there that cannot synthesize their own cholesterol, so they have to get this only from their diet. What this means is that for a lot of animals, it's just not possible to make myelin, or it's just too expensive in terms of materials. And this is the case for a lot of invertebrates. A lot of invertebrates don't make myelin. So what are the alternative solutions to increase conduction velocity instead of just myelinating it? And the other thing that you can do instead of increasing the resistance of the membrane with myelin or something like that is to increase the axon size. So this is something that, for instance, cephalopods use, and earthworms and a few other things. Earthworms only do this with a couple axons, but cephalopods use this fairly extensively, is that their axons are greatly, greatly increased in size, in diameter specifically. So if you look over here to your right, that's actually an actual photograph of a squid, specifically loligo axon. That's a really large cell, and they use these really large cells to conduct these impulses, these action potentials down these axons at a very high rate of speed. Now, it does not get to the same speed that we get with our myelinated axons, but it does do a lot better than a standard invertebrate axon.